Good morning, everybody. I hope you can hear me loud and clear. Welcome to our webinar for today. So we'll just give people just a minute just to get settled and stir the sugar into their coffees and set up their laptops and PCs for maximum comfort level. So whilst people are getting settled in, just take a moment and we'll be starting imminently. So good morning, everybody, or indeed good afternoon, depending on where you're connecting from. Everybody is welcome. I'm hoping today is as a lovely sunny day as it is here in the south of the UK. So welcome on board for today's webinar. We'll look to take approximately about an hour of your time this morning to look at alternative methods in the food business and how to approach that. And the title is indeed Trusting Alternatives, Unveiling ISO 16140 Part 2 for Reliable Method Validation. So I'm hoping that is a topic that is piquing all of your interests. Before we roll on, I'm sure you're all wondering, who is this guy sitting in front of you all? It's my pleasure to be with you today. I am your host. My name is Rob Butler. I am marketing manager here at bioMérieux for all of our food diagnostic tests and services, instrumentation and everything in between for Northern Europe. I'm based in the UK and I've been with bioMérieux now for I think around 20 years. I think I stopped counting at about 17, but I'm pretty sure I'm due another day's holiday pretty soon for my 20 years service, so something to look forward to. And indeed, I'm representing bioMérieux, and it is our pleasure to be co-hosting this webinar alongside bioMérieux with Campden BRI. And with that in mind, you are here to listen to our two illustrious speakers. So we have Suzanne from Campden BRI and David from bioMérieux. So Suzanne will be up first, and she is section lead for molecular microbiology and methodology at Campden BRI. It's been there since, or performing that function since 2005, and indeed is an expert and heads up a lot of the third-party method validation for alongside various entities. So I think that makes her rather qualified to talk on today's topic. Also, an active member of microval technical committees for certain ISO activities and indeed ISO 16140. Researcher and consultant on molecular methods for the food industry at large, and is an expert PhD supervisor for gut microbiota research, which is obviously a very topical subject at the moment. And indeed is very actively involved with extensive experience at European research project level and molecular techniques indeed. And Suzanne will be talking to us on the ISO 16140 Part 2, how to approach it and what to consider for that itself. And then indeed, for the second part, we'll be hearing from David Tomas, our Global Scientific Affairs Director for bioMérieux for all of the EMEA region. And he will be then taking a look at perhaps a case study and how we can apply that type of approach to alternative methods, and in this case, perhaps PCR methodology. So indeed, David is very active in coordination of Spanish Working Group on Standardization in Food Micro. He is also indeed a delegate and expert in ISO and CEN international activities and various committees that he will sit on within the food business. A member and research institute of the food engineering at Universitat Politecnica de Valencia. Sorry, David, I know you're wincing at my my pronunciation there. And indeed has worked as a technical auditor for the Spanish accreditation body ENAC, senior scientist for Nestle Research previously, and manager of the microbial and molecular laboratory in Enya, Spain. So a very warm welcome to our two great speakers. And without further ado, I would invite Suzanne to take the floor and begin proceedings. Thank you very much indeed.
Thank you, Rob, for the kind introduction. I will just share my screen. Can everyone see the slides okay?
Looks good to me.
Okay, thank you. A warm welcome to everyone today. And my first presentation today will be looking at microbial methods. So as food microbiologists, we have a choice of a broad range of methods for analysing our samples. And it's important that we choose a method that's right for us and to meet our needs. And one of those important deciding factors will be the performance of the method. And one way we assess that is through formal validation of ISO 16140. And this morning we're going to be exploring method performance and how we evaluate that. Okay, so the discussions this morning will cover 3 points. The first will be looking at alternative methods and the advantages that they give. We'll also be looking then at how alternative method performances can be assessed and what evaluations are available for alternative methods. I believe, Rob, there's a poll.
Indeed, there is. Thank you for the cue. The cue for the cue. So the poll is, how familiar are you with ISO 16140 method validation? So hopefully you see that prompt in front of you, and I can see some results coming in quite actively. So either new to ISO 16140 method validation, so a complete, let's say a novice, I'm aware but looking to learn more, so perhaps a little familiar but room for a bit more improvement, and those let's say experts out there that are here perhaps to judge us on our knowledge and comprehension rather than anything else. So results coming in, I think they look to be stabilizing. Voting has ended. So I don't know if people can see those results, but we have about, so 30% are new to, so let's say rather new to ISO 16140, so perhaps a lot of room for learning there. 2/3, about 68% are moderately familiar, but definitely looking to learn a bit extra, and only 3% are very familiar. So there we go. 1/3 to 2/3, pretty much, newbies to intermediate.
Okay, thanks very much, Rob, and thank you everyone for sharing their experiences. So moving on now to the rest of the presentation, and just wanted to start with a definition of what we think of as an alternative method. And this is actually taken from the 16140 Bible for method validation in the vocabulary section. And the alternative method is defined as a method of analysis that detects or quantified for any given category of products the same analyte as is detected or quantified by the reference method. Okay, so we've defined an alternative method. What are the benefits? And there can be several benefits. The first and perhaps most forefront would be the time to results, so much more rapid than the reference method. Potentially, they can be more sensitive, particularly for molecular methods. They can offer perhaps more sensitivity than cultural-based methods. They're often easy to use, and that's one of the reasons why labs will implement alternative methods, is that they are much more straightforward, perhaps in setup or reading of the results. There's also the potential for automation. So with laboratories looking perhaps to be more efficient, automation might be the way forward for them. They're also a standardized protocol through the kit insert and QC checked reagents. So that saves time in terms of potential checks that need to be carried out within the laboratory. And also that their performance has been usually assessed by independent external validation. Therefore, there's a third-party assessment of performance, which is often useful when deciding how to pick your method. So there are a range of different methods that can be used within the food industry. The first is the cultural methodology, and that may be perhaps through chromogenic media, which gives better differentiation of the target organism that we're looking for, or potentially optimized enrichment broths, which reduce the time for the initial enrichment for particularly pathogen detection methods. We also have molecular-based methods, so polymerase chain reaction or PCR, and that tends to be the predominant methodology, particularly for things like pathogen detection. There are also some newer methods looking at isothermal amplification, where the DNA detection happens at a single temperature, and we're seeing more of those coming through in the last few years. We can also use immuno-based or enzyme methodology, so ELISA, which is a methodology that's been around for quite a period of time. There's also lateral flow devices. I think most of us are familiar with those through the COVID testing that we've done in the last couple of years. And also immunomagnetic separation. And this can be very useful for pathogen-based methods, particularly when the pathogen is available at a low level of contamination. It's able to perhaps concentrate those organisms and potentially remove any impurities, either for a cultural step or potentially a molecular PCR-based detection. There've also been, in the last couple of years, some instrument-based methods, particularly those for detecting indicator organisms and also commercial sterility assays. So as a microbiologist, we have quite a lot of requirements for our methodologies. The first would be that it's rapid. It's an internationally acceptable method. It's robust and is able to cope with the types of samples that we will be testing. It's reproducible, so produces consistent results. It's easy to use and implement in the laboratory. There's a standardized protocol and also reagents available for analysis. There's defined guidelines for interpretation and also defined criteria for assessing competency. Okay, so we've talked a little bit about alternative methods. Let's just explore now method validation in a bit more depth. And again, there's, as you would expect, a definition within the 16140 protocol. So for validation, it is the establishment of the performance characteristic of a method and provision of objective evidence that the performance requirements for a specified intended use are fulfilled. So what can we actually use those validation studies for, and why are they important? Well, the first thing is that we can use them to demonstrate equivalence of any commercially available methods for the analysis of products named within the EU microbiological criteria EU 2073, and we'll talk about that in a little bit more detail later. We can also generate results to confirm that the method is fit for purpose. Now that's important for accreditation, particularly to standards such as ISO 17025. And also the validation studies will provide some results which we use as reference values for method verification work. And that's following another standard within the ISO family, ISO 16140 Part 3. Okay, so why is method validation important? Well, we need to, within Europe, we need to comply within the European law. And for food businesses operators, they will have, particularly with ready-to-eat foods, criteria that they need to meet. And also within the standard, we also have the method which is named to be used, and that's typically the ISO reference method. So if you do want to use an alternative method, there's actually some further details within this particular criteria. So it is acceptable to use an alternative method, but it needs to be certified by a third party in accordance with the protocol set out in ISO 16140. So you can see there's a nice tally-in with method validation to prove that the method that you're using is fit for purpose for the types of samples that you are testing. Okay, so there's two terms that we tend to use with method evaluation. The first one, which we've already talked about, is validation. And the second one is verification. And this is just to do a comparison, side-by-side comparison, to look at how they differ. So the validation is a study which proves that the method works and detect the target organism, whereas verification is carried out by the using laboratory, and it's to prove that they can actually use that method and give the correct results. And both of those definitions again are taken through by the 16140 definitions. Okay, so we've talked about method validation. What's the benefits of those? So we've previously mentioned that it's an independent assessment of method performance. We have, with ISO 16140, we have defined protocol requirements and also assessment criteria. So everything is done consistently and following the same rules. We compare our methods against standard recognized reference methods. Again, that's really important because we're using a gold standard method and one that can be used as a very effective comparator. And also with third-party validation, it's peer-reviewed, both in terms of the protocol and also the data analysis. So we have technical experts that are reviewing the data to ensure that the validation has been carried out correctly, but also that the results meet the required criteria. So we've talked about validation. What sort of methods can be third-party evaluated? So there are a range of different methods. So we can look at enumeration methods, detection methods, semi-quantitative methods, which are able to detect a particular target organism at a defined threshold. And then also confirmation and typing. So perhaps towards the end of your detection method, you want to have a method that is good for detecting and confirming your target organism with good accuracy and effectiveness. So as well as the methods that we can currently use, ISO 6140 is also developing up some methodology and protocols for the detection of viruses and parasites, and as well as the quantification and detection of bacterial toxins. So we're still very active in this area of method validation. So we've talked about method validation, what organizations actually carry out the validation studies. So we have a split between both Europe and the United States. So within Europe, we follow the ISO 16140 Part 2 criteria, and there are 3 organizations which carry out those studies. There's Microval, which is throughout Europe, Nordval, which tends to be focused in Scandinavia, and AFNOR, which is based in France. And then within the States, we have 2 types of analysis. So typically, most of the methods will go through the performance tested program or the PTM program, but there's also available the official methods of analysis, which is a little bit more of a stringent study for assessing method performance. So there are differences in the protocols, but we are working currently to harmonize those protocols to make sure that we're getting them closer to each other. And in fact, there are new standards for both Europe and the States where we're looking to try and harmonize those protocols further. So if you're interested in method validation and the third-party organizations, each of those third-party organizations have a website that's available. And with these, you can access and search for methods that are approved by these organizations. And they also have available on their website for the approved methods, the certificates that are available, and also for the validation reports. That will give you a lot more information on the performance of the methods and the types of food matrices and samples that have been tested. Okay, so we've talked about just in general for method validation. So a little bit more about the studies and what we need to carry out and the study essentials. So it's a method study comparison protocol. Typically, we're comparing the reference method, which will be the ISO reference method, to the alternative or candidate method. And within that protocol, we have a defined procedure and assessment criteria to carry out that assessment. And along with the 16140 Part 2 protocol that we need to follow, we also have a companion in Part 1, which gives a glossary of terms. And those are the definitions which I discussed with you earlier, plus many others to actually look at how the validations are carried out. So what sort of things are included within the validation study? Well, the first one will be the scope of the analysis, so the foods and samples that we need to analyse. And that can either be a broad range of foods or it can be selected categories. There's also details of the sample weight, which was used for the analysis study, and also some details on the sample preparation. Typically, that will follow the ISO 6887 series, but there can be the option to use different diluents, different dilution ratios, and this is where the protocol will take into account those adaptations, particularly when we're using different enrichment media for the reference and the alternative method. There will be the details of the reference method, which is typically the ISO protocol, and also details of the alternative method as well. So there'll be a defined protocol, and also all of the details that we used in the study will be listed within the kit insert. Okay, so I've talked about the types of foods and food samples that can be analysed during the study. So how do we define categories? Now, these are groups of foods that share similar properties, and there's an example below of a particular category which is listed within the ISO 6140 criteria, and this is for heat processed milk and dairy products. And for our evaluation studies, we need to pick a selection of 3 food types. So if we were carrying out an evaluation with this particular category, we'd have a choice between pasteurized dairy products, sterilized or UHT products, pasteurized milk products, milk-based products, or dry milk products. So you can see, depending on the types of foods that you're analyzing, and also the potential pathogens that you are or organism that you are detecting or enumerating, you may vary the food types that are included in your evaluation. And there are 15 food categories that are listed within the standard. And we also have some additional categories, and you can see this on the next slide here. So these are the full categories that are listed within the ISO 16140 Part 2 standard. So all of the food categories are listed in green. And then you'll notice at the bottom on the right-hand side, there are 3 different sample types that are highlighted in yellow. So these are the additional the additional sample types that we can include within the evaluation study. And these are pet food and animal feed, environmental samples and food or feed production, and also primary production samples as well. So we've talked a little bit about how the studies are designed. How do we actually evaluate performances? This table here gives some details on the qualitative studies, and you can see that we assess a lot of the important factors that we look at when choosing a method. So the first one would be, is it specific? And here we want to check for the potential for false positive or negative results, and that's included in an inclusivity and exclusivity panel. And we also have a sensitivity study where we test a wide range of food samples to potentially look at interference from background flora, or potentially some of the food matrices as well. We want to make sure that the method can cope with a wide variety of samples, so it's robust. And this potentially can be assessed again within the sensitivity study, and also during the ring trial or inter-laboratory study. We look at the sensitivity, so we look at, particularly for pathogens, we're looking at the ability of the method to pick up low levels of target, particularly around 1 to 2 CFU per test portion. Again, that's tested within the sensitivity study where we use artificially contaminated samples. And again, we want to have a look at the agreement with detection between the reference and the alternative method, so the amount of trueness between the 2 methods. And again, we assess that within the sensitivity study. So for our limited detection, we assess that within the relative limited detection study, and also the LOD50 study. And for those of you that are more familiar with method verification, LOD50 will be part of the analysis that will be carried out within that verification study. So we're looking at the lowest level of contamination that can be detected. And this can be matrix dependent, and that will need to be taken into account during the method evaluation study. And then we also want a method that's reproducible to give consistency across the the laboratories when they start implementing the method. So typically the first part of the study, so the inclusivity, exclusivity, sensitivity, and relative limit of detection will be performed by the expert laboratory. Whereas the interlaboratory study is the opportunity for other laboratories potentially that will be using the method to take part in a ring trial to assess the performance of the method. Okay, so we've looked at qualitative study. Again, there'll be a similar sort of matrix, evaluation matrix for quantitative studies. So again, we'll be looking at specificity to target with the inclusivity and exclusivity study. We want to look at the accuracy and the precision of the method, and that will be looked at during the accuracy profile section of the method. We want to look at any potential biases between the reference and the alternative method, and also the ability, again, that the method is robust and capable of coping with the wide variation of samples that will be tested potentially during an evaluation study, potentially by you as method users in the future. And that will be looked at with both the relative trueness and interlaboratory study. And for reproducibility, again, we'll be looking at the ring trial or the interlaboratory study. So in summary, there are many demands required of analytical methods to meet both safety and quality requirements for testing laboratories. There are a wide range of commercially available alternative methods, and they can offer the laboratories many benefits. But alongside that, there is the responsibility of the laboratory to choose the method that is right for them, that meets the requirements of their customers, any potential legal requirements, and also any requirements in terms of the matrices that they're testing. And with the evaluation protocols through ISO 16140, there's a robust evaluation that is in place to evaluate methods. And that's available through the websites. We are an expert laboratory for AOAC, Microval, and also Nordval. So we carry out some of those studies for alternative method manufacturers. So just we've covered a lot of method validation. The final part when laboratories are looking to implement a method within the laboratory will be checking that they can actually use the method and it gives them the results that they're looking for. And there is another part of the ISO 16140 family which gives the protocol for doing that, which is 16140 Part 3. And there are currently protocols in place for qualitative, quantitative, confirmation, and typing methods. There is a clear and defined protocol and criteria, which is really useful for laboratories because it gives them not only the number of samples that they need to test, the types of levels of contamination that they'd be looking for, but also some evaluation criteria as well. So everything is kept consistent in terms of method evaluation. So the validation study in combination with the verification study would be a good source of data for laboratories that are planning to submit any methods for ISO 17025 approval. It also potentially will give a lot of information on staff competency and also instrument qualification. And then down the bottom of the slide, if anybody is interested, there's some information here on the ISO website, which gives some further information and also spreadsheets that can be used for laboratories when they're carrying out method verification studies. And I believe, Rob, that there's a final poll coming up.
Entirely possible. Yeah. Apologies. I think I jumped the gun a little earlier there. So hopefully that poll is appearing in front of your screens now, everybody. So would you be looking for further support on method validation or indeed verification? Options being more in-depth training on method validation, method verification training, support on validation study design, or at the moment, probably not. Answers coming in still actively. Looks like people are still voting. Few more seconds. Oh, And I think perhaps we can call it a day there. So a mix there. So not at the moment. So perhaps people maybe not quite ready to look outside of their own organisations for support at the moment. But 12% saying more in-depth training on method validation would be of use. 28% saying method verification training could be useful, and 17% for validation study design. So a bit of a mix there. Interesting.
Yeah,
Thank you very
thanks, Rob.
much.
Thanks, Rob. And then I just want to conclude the presentation by thanking everybody for their attention and look forward to questions later on during the session.
Wonderful indeed. Well, thank you very much indeed, Suzanne, and very timely. So hopefully you all found that pretty enlightening. If you do have any questions or comments indeed, please do not hesitate to use either the chat functionality or submit your questions. So there is a dedicated function for that in the top right hand corner if you see that. So you can put all of your questions for either speaker or indeed both up there, so please don't hesitate, and we will look to consolidate those and have a bit of a question and answer and general discussion at the end of the two presentations. So moving swiftly on, David, I see you're there and ready to go. So David, I would invite you to the floor. It is all yours.
Okay, perfect. Thank you very much. It's okay, the screen like that?
Yep, looks good to me.
Hopefully, yet. Okay. So the idea during the other part of the session is to present you a practical approach. So indeed, I'm going to present all that has been already presented by Suzanne, but showing some practical examples. So, and I will focus on an alternative PCR method for pathogen detection. That is one of the technologies that can be validated to have quick results for your laboratory. So the idea is first a short introduction because Suzanne already presented the ISO 16140, but as 30% of the audience is not familiar with this standard, I think it's good to show a short introduction, and then the case study on the validation itself and some conclusions. So let's go for the introduction. As already Suzanne mentioned, the ISO 16140, it's a series of standards on method validation. So you can see here that starting by the one that is the vocabulary, and then I jump it to the 4, 5, 6 that are already published. So you can buy it and you can use it for single laboratory validation, for factorial interlaboratory validation, and for alternative confirmatory and typing protocols. And then new parts are now in development, in the pipeline, that are identification methods, viruses, parasites, and also bacterial toxins and biogenic amines. So all these are coming, but for this specific presentation, we already mentioned the part 3 and the part 2. So the part 3 is used by all laboratories that wanted to implement a method, and the part 2, it's mainly used for the manufacturer, for the people developing methods that are selling the methods to the users. So green for the users, red for the manufacturers. And in all the standards, we also have this kind of flow chart that, as you can see, red, it's for the manufacturer. So if the method is not validated, then the requirements legal in Europe are requiring to validate according the part 2. So you need to choose the part 2. So also we have several parts. Only the part 2 is legally accepted in the European Union. And if you are going to verify the method in your laboratory, you follow the green box. So you need to use a method already validated. So an alternative method validated. So you only can do your verification in your laboratory according to the part 3 if the method has been, if the alternative method has been already validated. So if no validation for the method, you cannot do your verification. And that's, for me, important to highlight. That is a criteria that you must take into account. So only alternative validated methods can be verified and implemented in your lab. So going to the case study, this is an alternative PCR method. It's called GNAP Salmonella. You can see here is a thermocycler. And then the method protocol simplified is on the right. So you can see that it's a same method for all matrices. So the DNA extraction, it's very simple, one pipetting, and then you include the samples into the PCR, and in less than 1 hour run, you will have your positive samples that must be confirmed. So you can see that all these steps are common, but the sample preparation and enrichment steps include several options. As Suzanne mentioned, we need to validate all options. So we have a protocol with 37 degrees, another protocol with 42 degrees, another protocol for swabs and sponges for environmental samples. So then we need to be sure that all the sample preparations protocols are included in the validation study. So let's then, and also we have some specific validation protocols with this automated DNA extraction to reduce dramatically the time to results. So all those must be included in the validation. So let's go for the validation. And I think you have a poll to understand if you are familiar with the validation has been launched, Rob?
Yeah, I think we do indeed. So hopefully that poll is on your screens now, and I see some answers already coming in. When using alternative methods, have you checked the validation data available? Answers being, I only check the method validation certificate, so paperwork exercise. I check the validation certificate and the report itself. I'm checking only the method protocol, nothing further than that. And I do not use validated alternative methods at all. So perhaps irrelevant at this stage. Answers still coming in. It's just a few more seconds, but I think we have a bit of an overwhelming answer, and I don't think it's a big surprise. Okay. Right, poll has ended, and well, we'll have to trust you all that you are the good students that you're claiming to be, with 75%, so 3/4 of you checking both validation certificate and the report. Some limited only to the method validation certificate, and then a few very limited that are either only checking the protocol regarding the methodology, and a few that at this stage it's irrelevant to.
Okay, this
Thank you
means
very
that
much.
I don't need to present anything because all people is familiar with that. But let's go through the different, but I can be quick in some aspects considering that you already have experience reviewing not only the certificate, but also the validation protocol itself. So let's go for the example. So as Suzanne mentioned, we need to select a reference method. And for Europe, the reference method included in the regulation, for example, for Salmonella is the 6579 part 1 published in 2017, but also we need to consider the amendments. So in this case, for example, we include also in the validation the amendment from 2020. So this is the method that we need to compare with. This is for us the reference, the gold standard. Then we need to select matrices, the sample to be tested during the validation. And of course, we want to cover matrices worldwide available from all different food categories. That's the original idea. But in a validation study, it's not possible to cover all the world and all the food matrices. So we need to arrive to a compromise. And let me take some time to explain because I receive a lot of questions around the scope. So uh, the method scope, for example, in this case, for the reference method, it's available for a broad range of food. So it's an horizontal method for broad range of food, for feed, environmental samples, and primary production. And remember that Suzanne mentioned that those three are additional categories. So for the validation, you need to address these three categories on top of the broad range of foods. So you can use the method for all foods in the world. But the validation study is not going to include all these kind of foods. So the validation scope, it's required to include 5 food categories to cover a broad range of foods. So if my validation study includes at least 5 food categories, I can claim broad range of food, and you, the laboratory user, can use the method for all range of food. So you can test some food categories that are not included in the validation scope, but are included in the method scope. So for this specific case, the method is validated for a broad range of food because more than food categories were included in the validation. And also for this specific method, it was validated for feed, for environmental samples, and for primary production. So if during the validation, you don't include, for example, primary production samples, you cannot claim for that. And you need to consider that the method scope is not covering primary production. Okay, I hope this is enough clear. And then the use of the laboratory, of course, can include some of the matrices included in the validation scope, but also some others that are not included in the validation scope, but the method is valid for those matrices. You cannot work outside of the method scope, but you can test matrices that are not in the validation scope. I hope this is clear. Then we go to the instructions for use. So very important also to have a look to the instructions for use of the method. So I copy paste it here, for example, for the method, and you can see that the method is certified in this case for AFNOR, but can be microbial or other. You have here the certificate, and then you in the method is included all the protocol that are validated and all the sample preparation protocols validated, including the sample size, including the temperatures, and including the time to results, the time to incubate. So you can see that we have standard protocols, but we have large size protocols for chocolate, for example, and then we have a number of different protocols for milk powders, large size, so up to 375, pet foods up to 375 grams, primary production samples, so you need to use a different enrichment broth, or large sample size for chocolate up to 375 with a protocol that gives you results in 10 hours and requires the use of this automated extractive platform. So with that, it's important to highlight that please check always the instructions for use to be sure that the protocols and the sample preparation protocols that you are using are included in the validation and has been validated. So once you have the reference method checked and the scope, then we move to the certificate itself. So this is a certificate, in this case, again, from AFNOR, and the certificate includes a number of information. So the certificate, in this case, is a 2-page certificate, and you need to have a look on a specific points, and I highlighted. So those are, in my opinion, what is critical for you to be applicable in your laboratory. So let me highlight this. So you need to consider if the certificate is valid, because you need to renew, you need to send again the certificate every 4 years, in this case, for renewal. Again, you need to check that the reference method is the method that you require. So this method has been validated against the ISO method for Salmonella, and we can claim equivalent results compared with the Salmonella method ISO, and including also the amendment. Then, important for accreditation, the traceability to the version. So you need to follow the instructions that are mentioned here in this number, and also the version. So we have several versions that are included, are validated, and each time that we prepare a new version of the protocol, because we have a change, we have adaptation, we have an improvement, we need to submit this to the certification body to get the agreement on the new version and to be included in the certificate. And the fourth thing that you need to have a look on the certificate is the scope. So here we have all human foods, the temperatures, pet foods, environmental samples, primary production samples. So you can see here that the method covers the food that you are going to analyze. So this is important for the certificate. As you mentioned, most of you are also checking the report, the validation report. That is another document. Both documents, certificate and validation report, are public and fully available in the respective websites. So you can download it and you can save it and you can check what, the last version available in the website. So the validation report in this case is performed by this expert laboratory, but as Suzanne mentioned, Campden is also expert laboratory, not only for the ISO 16140, but also for AOAC. But in this case, it was performed by this expert laboratory. And then you have a report that you can here include almost 400 pages. So for all of you that say, I'm familiar with the report, I can see that you spent a lot of time looking at the 400 pages of the report in this case, for example. So I'm trying in the next 15 minutes to summarize what is important to avoid you to going through the 400 pages, but still are available. But I will try to highlight what is most relevant in a few slides. So first, the history of the method. So as I mentioned to you, the first validation of those methods was in 2016. You can see here the initial validation study. And then the method was extended or modified in different periods until 10 extensions. Last was December 2023, so December last year. So at the very beginning, you should have all the historical changes of the method, because you need to consider if these extensions or modifications are impacting your method, and then you need to implement those changes in your protocol. So have a look always to the historical data on the method. Then we move to the method itself, and I'm going to talk only for qualitative detection method, and those are the minimal requirements included in the ISO 16140. So as Suzanne mentioned, we have two steps. One is an intra-laboratory study performed by the expert lab. And then you have also an inter-laboratory study performed by minimum 10 laboratories. So at the end, you need to do several studies, including a minimum number of samples to claim the broad range of foods. For example, if you 5 food categories plus the extra 3 categories if you want to claim that in your method. So this includes 300 samples, 150, and then inclusivity, exclusivity, and the sensitivity. Let's go for the data included in the report. So the first is the sensitivity study. So here you can see sensitivity study. I'm going to talk about these two studies. Sensitivity study, the objective is to assess what is the method behavior in a very broad range of foods. So we try to cover as much as possible all foods included in the market. So that's the objective, to see what is the performance of the method with a big number of foods. Of course, we want to include as much as possible natural contaminated samples. And that's in this study, for example, only 5% of the samples were natural contaminated with salmonella. That maybe is a limitation for the validation study, but it's a good number because it shows that salmonella is not broadly available in the market. So we were not able to find a lot of natural contaminated salmonella samples in the market. So that's good for the food producers. For the validation study, if we don't find enough natural contaminated samples, we need to spike. And usually 50% of the samples are spike or positive, and 50% of this sensitivity study are negative. So for this specific study, you can see that the minimum was 5 food categories, but we tested up to 20 different sample preparation and food categories. So it's including more than 1,600 different samples. Remember that it was required at least 300 for broad range of food. So you can see the extension of this study covering a big range of foods, and you can check in the validation report. What is the outcome of the validation sensitivity study? Well, the outcome is a performance characteristics. So I recommend you just go to the table with the summary of results. In this case, it's the 12, can be a different number, but there is always a summary results of the sensitivity study. So here you have the sensitivity for both the alternative and the reference method. Sensitivity is the ability of the method to detect the target, to detect the salmonella. So here you can see that the sensitivity for both methods are more than 95%. This is the consumer risk. If you have a low sensitivity, means that you can have salmonella in your sample, not detected by your method. The thoroughness is the agreement between both methods, positive and negative results. So you can see that in almost 98% of the cases, the alternative method and the reference method are giving same results. And then you also can find the false positive ratio. This is the producer risk. So in 2.6% of the cases, the method can give a positive that will be not confirmed. So you are not confirming the method at the end, but you need to confirm. So it takes some time to go for the confirmation steps with no need, okay? And then you have also the table with the discordant results showing if the method is not matching, what is the impact. So this is evaluated by the committee, and then you can see that here the acceptability limit is 3, for example, for this category, and the method includes 0. So the method is below the acceptability limit, meaning that is okay. The method is giving equivalent results between reference and alternative method. So, and then the summary. So for the sensitivity study, just one table and one disclaimer that is saying that the method is met the acceptability limits. Let's go for the next data that you can check from the report. That is the relative limit of detection. Here, we are not checking a lot of different food items, but focusing on the different food categories and selecting one food item for each food category. Again, in this case, we need at least 5 for a broad range of food. Here, there are 20 different RLODs. And what is an RLOD? Is the comparison between the limit of detection of the reference method and the limit of detection of the alternative methods. And again, the ISO established acceptability limits, and then you need to meet these acceptability limits. If you have one, means that both methods have the same limit of detection. Limit of detection is the minimum amount that you can detect of the target of the salmonella. So here, usually the information is very limited because you need to pass. So don't worry, you don't need to check every single figure because you need to pass. But as Suzanne mentioned, for the laboratories, for the users, you need the level of detection. So for method verification, and then you need to check just the column with the alternative method. Those are the values that you need to verify your method. See, if you are familiar with the part 3, you need these values for the protocol 1 and 2, not for the protocol 3. Okay, but I'm not going to explain verification now, but is needed for protocol 1 and 2 of verification. So again, here you have the CFU per sample size that the method is able to detect in 50% of the cases. And then this is the value that you need for your method verification. I'm not going to talk about inclusivity, exclusivity. Usually you need to pass, so the report are always including this data. And to finalize my part is the interlaboratory study. So what are we looking for with an interlaboratory is to see if the method works well in different countries, different laboratories, different conditions. So we need at least 10 different laboratories. In this case, we have more than 10 laboratories that participated. So, and you can see that you have a organizer, that is the expert lab, distributing samples for all, in this case, Europe, that can be done in US or in different. So there are requirements in terms of different countries, different organizations must be included in the interlaboratory study. So, and what is the outcome of the laboratory study? Is again, sensitivity, relative trueness, and false positive. So in this case, for example, all samples were confirmed. And then you see that you have a good agreement between the alternative method and the reference method, and also the limit of detection was equivalent for both, for the reference and for the alternative method. Here, we only test one food item, one food category, because the idea is not to test again a number of foods, because we already tested in the previous study, but just to see if the laboratories are able to perform the method in different conditions. So to conclude my presentation, the validation according ISO 16140 Part 2 is crucial to demonstrate alternative method performance and compliance. Without this validation, you cannot verify the alternative method in your laboratory, and you are not following the European regulation. Then the certificate and the report provide relevant and useful data, and only with 3 tables you can have a look on what is the general performance of the method and the method benefits, and why not the method limitations, because in all methods we have some limitations. And then again, the validation must be complemented by the method verification. So the laboratories must use the 16140 Part 3 to verify the already validated method. That's all. So if you have questions, thanks a lot for your attention. And if you want also, of course, to contact me or your bioMérieux representative for more clarification, you have here my email. So thank you very much.
Wonderful stuff. Thank you very much indeed, David. So thank you to both speakers. Moving very quickly on, perhaps just a couple of minutes to address some of the questions that were coming in, if you will indulge us. So Suzanne and David, cameras are back on. So I'll dive straight in the interest of time. So I do have a question regarding matrices. How are these picked for the validation studies? How does that actually come about? Perhaps that's one for Suzanne.
Okay, so we talked a lot about categories, food categories, and we mentioned that you need to have 5 categories to fulfill a broad range of foods. And within the ISO 16140 standard, there is a list of categories at the back and the types of organisms that you would associate with those particular food types. So it's really important when we're choosing the categories that it's relevant to the particular organism. So for example, with salmonella, which we've discussed today, we would potentially include poultry samples in that, because we know that potentially they've got a high incidence of salmonella, potentially salmonella contamination likelihood. We might look at other products such as dairy, so we want to spread the types of organisms to perhaps different microflora that might be present, perhaps different fat contents. So yeah, it's usually quite a strong debate looking at particular food categories that will be involved within a study, but it is just to give as good a balance as possible for a method that gives a broad range of foods samples. If you're looking at the 3 matrices that we discussed, so the environmental samples and the primary production, and also the farm and feed category, those are separate. But yes, it's always tailored to the type of organism that we're looking for.
Makes sense. Yep. Very good. Hopefully that answers the question. I'm just looking through. I don't think we'll get through all of the questions, so we might run a second or two over. We did have, so PCR methods. Will there be PCR methods in future that do not include the isolation of the target strain, as this can be a bit of a constraint when in official controls there is no isolate for further epidemiological research? So do we need to isolate that target strain?
Today,
I guess perhaps for
yes.
confirmation purposes, I guess. So yeah, David, perhaps.
Today the validated method includes a confirmatory step. It's mandatory. So today to validate the method, we need to run the confirmatory and to isolate. And from this data is coming the false positive ratio. That is a positive from the screening, I would say, or for the PCR or ELISA or whatever you are going to test, or the cultural method that has not confirmed. So today, yes. Having said that, in the new 7218 that was published in 2024, we have a text saying that you can confirm a method with another method. So now this possibility is open. But then for regulatory purposes will be not valid, but perhaps we can open the door to say, well, if you have, for example, with another PCR or an ELISA, then if it's the same enrichment broth, you can arrive. But today, this is not a possibility yet.
Okay. So we'll watch this space. But yeah, it's a fair question. It is a challenge. Another one. So Salmonella reference method is only validated for maximum 25 gram samples. In which case, how do you compare to an alternative method wishing to use up to 375 grams? I'll leave that question open. I'm not sure who's best. If
Suzanne, you can reply.
either of you wish to weigh in on that.
Yeah, I mean, usually the reference method will be validated for 25 gram samples. And if you do want to validate a method with a larger sample size, it is possible to compare a 25 gram sample with a reference method with the 375 gram reference method. If a laboratory did want to compare like with like, so the same sample size, they would need to prove that they can, or they would need to verify the reference method that they can actually handle 375 gram samples. But probably worst case scenario would be a 25 gram sample for a reference method versus a 375 gram sample for the alternative method.
Okay, very clear. How should the user lab deal with changes to a reference method on which the alternative methods has been validated against? Is there a statement included in the revised reference method or validation report to state impact to labs, i.e. potential re-verification process to be done? Does that
If you
make
want,
sense?
I can reply on that. So I'm part of the ISO together with Suzanne developing reference methods. So I'm talking now from the ISO side. Yes, in the introduction of each reference method is mentioned if the changes from the previous version are major or minor. So if the changes are considered minor, you don't need to re-verify. You don't need to do anything, just anything in terms of verification. You need to adapt the method, but not to re-verify. If the changes are considered major changes, then you need to re-verify your method against the new version. Why are considered major? Because the performance characteristics of the method are different. And then because the performance characteristics, maybe the limit of detection, maybe the sensitivity are different, you need to re-verify the method. So those are the main let's say changes on the reference method. What are a typical major change? If you change, for example, enrichment broth composition, temperature for enrichment broth, isolation agar, or different conditions from the reference method in terms of time temperatures. That's the typical major changes.
Very good. Thank you, David. Perhaps just time for one more. So according to the list of matrices validated, verifications included for a given method, what about if there are different than new categories of food that I guess perhaps didn't exist at the time, such as things like plant-based foods and other things that are coming forward? How do we approach that? Is that one clear? I think that one just dropped in just now, so you might not have seen that one on your feed.
I can start to answer that one. At the moment, we have the categories with a broad range of foods, and the plant-based, as you're right, are not included within that. But we do, at the moment, we recognise at ISO level that there are new sample categories that need to be included, and the plan is to include those in the next revised version of 16140 Part 2. But we do, within the verification protocol, you have the option to look at, you need to verify the method initially. So you have to use one of the food categories and samples that were included in the initial validation study. But you also have the option then to, with your additional food samples that you would test within the item verification, that you could then look at potentially plant-based foods within that. So particularly if there's a challenging category that you want to include, the recommendation would be to include that within your verification so that you can show to both your customers and also auditors that the method is fit for purpose.
I see. So perhaps within verification, you have the scope to be able to do that currently, and then for validations, they have to be enrolled into further waves done by the relevant authority. Very good. Well, thank you very much indeed for the questions, and indeed thank you both for your fielding those questions and your answers to those. To anybody that did pose any questions that didn't get addressed, we will be sure to follow up. So thank you indeed for all of those questions, and very sorry that we didn't quite have time to cover those all. If I may, just in concluding comments, if I can just share my screen. If you can just indulge me, just a couple of dates for your diaries that I'd like to make you aware of. So bear with me. So hopefully you can see my screen there. So just a couple of dates for your diaries. So we have Campden BRI will be holding their annual Hot Topics event, one not to be missed. So a conference normally towards the end of the year, and indeed this year it will be held on the 10th of October. I believe that is a Thursday. If it's not already in the chat, I believe a link will be posted in the chat that you can click on for registering and to peruse the agenda. So please do take a look at that and sign up. You'll see me there amongst many of my colleagues as well. And another date for the diary is a bioMérieux event. We'll be running a seminar on quality indicators, spoilage, hygiene indicators, and looking at all of that type of thing. Things like in-process controls, as well as early warning systems for manufacturing and that type of thing, and really how quality can help partner up with production to really bring value to the manufacturing chain. And we'll be holding that event. It's in Birmingham, I think in the Royal Leonardo Hotel in Birmingham on November the 20th. I believe that is a Wednesday. So please do mark that in your diaries, and we'll be communicating more on that and sending out invitations very, very soon indeed. So please put a placeholder in your diary. And final word is thank you to everybody. Indeed, thank you to our speakers for very interesting, very entertaining presentations. We hope that you in the audience also found those to be very enlightening. Thank you also to our audience for your participation and your engagement and your questions throughout. And we look forward very much to the next one. As a tiny bit of homework, I think as we close the session, there will be perhaps a small pop-up survey. It'll take you perhaps 30 seconds just to fill that, just to gauge your feeling on today's proceedings, how we did, but also most importantly, what you'd like to see in the future and how we can improve things. So we would very much appreciate your feedback on that so that we can come back bigger and better next time. And I think pretty much that concludes everything for today. Apologies, we ran over slightly, but hopefully that is a testament to the quality of the content. Thank you very much indeed for your attention, everybody. Thank you again to our speakers, and I wish you all a very wonderful rest of the day. Thank you all.
Thank you.
Thank you.
Bye.
Bye-bye now.